Hello, ladies and germs. Welcome to the music of insects. Or how to make friends and influence arthropods. With me, your host, Dr. David Bashwiner. Your inner insect. How closely related are we to insects? Well, it depends. We do all live on the same planet. We appreciate many of the same foods. We share lodgings, for instance, with roaches, termites, bedbugs. We have a common ancestor of about 600 million years ago. What, pray tell, was that common ancestor like? Probably like a worm. It was a bilateral animal. Most of the invertebrates and the vertebrates diverged from that ancestor 600 million years ago. It couldn't yet hear, or, of course, sing, nor could it breathe air or fly, but it did have a nervous system and some of the same basic structures of the brain that both we and insects still have today. This is a little picture that we developed last lecture, and we see over here that we have all animals, we have all bilateral animals, and over here diverging off are insects and other invertebrates, and then we have the vertebrates going here, and then we have a bunch of different types of vertebrates, and if you follow it all the way along, you would get us. And like I said, the humans don't have to be at the end of this. Humans could be kind of anywhere in here. You can draw this any way you want. But I made this as our own ancestry, so if you looked at us going back this way, you'd have to go back to about 600 million years ago, and that is when our common ancestor with insects would have been around. And we noted that lampreys don't seem to be able to hear, and they don't vocalize. Sharks probably can't hear also, and they don't vocalize. And then a bunch of fish species do, but they do it in different ways, so it seems like somewhere around here is when hearing and vocalizing evolved. And then all vertebrates after this point have the same sorts of mechanisms for both hearing and singing. But before this, there wasn't anything. So our common ancestor with insects wasn't able to hear or sing, but a bunch of different types of insects can hear and sing. They evolved that independently, and we believe that they evolved it 7 different times. Insects did hearing. They evolved hearing 7 different times. So, what does this all mean? Since our common ancestor did not hear or sing, we know that these capacities evolved separately in the ancestral line that led to insects and the one that led to us. So an interesting question is, do insects end up hearing and singing in the same way that we do, or did they evolve different means of doing these same things? What might be the same about how humans and insects hear and sing? What might be different about how humans and insects hear and sing? Now, I wanted to give you a diagram similar to the one we looked at just a page or so ago, and this is just looking at a different way. So organisms are just life forms, and you have unicellular and multicellular organisms. And it doesn't really completely divide this way, but prokaryotes are definitely all unicellular, and they diverged back here about 2 billion years ago. Prokaryotes and eukaryotes separated. So this is 2 different lines, and eukaryotes are the ones we're going to be talking about. This produces both vertebrates like us and invertebrates like the insects. So we don't need to know anything specific about this, but eukaryotes just happen to have cells that have specifically a nucleus inside them with little organelles. Organelles are like mitochondria and I actually don't know what else they might be, but little tiny organs inside each cell. And then eukaryotes are mostly multicellular, but there's certainly some unicellular eukaryotes too, but there are no multicellular prokaryotes. All right. Now, among the eukaryotes, there's the 3 main groups: fungi, animals, plants. You would think that fungi would be more related to plants than to us, but they're not. Fungi are actually much more closely related to animals than they are to plants. We're going to be talking mostly about animals today. Animals can be defined roughly as organisms that breathe oxygen. They consume organic matter, meaning they eat. They're mobile, meaning they can move around. Plants can't really move around as much. Plants breathe carbon dioxide and give off oxygen. So it's the opposite of animals. And then animals reproduce sexually. And some plants do that too. So about 650 million years ago, you get the origins of animals. And in looking up this, like a definition of animals, I read that among all living animals today, you have 6,331 groups of genes that are common among all of them. So you might think like, how close are we related to insects? Apparently we have at least 6,331 genes in common with all insects. Okay, so animals subdivide into two groups, the bilateria and the non-bilateria. Non-bilateria is not really a group, but the bilateria is a group. This happens about 600 million years ago. And so the non-bilateria are the animals that are not bilaterally symmetrical. So sponges, jellyfish. A sponge, you wouldn't think about it, but it's alive. The thing, it might not be the one in your sink unless you actually are using a sponge that someone got from the beach. But sponges that you get from the beach really do look like the things you use in your sink. They don't have a head. They're not divided in half. Same with jellyfish. So those are animals. They're just not bilaterally symmetrical. Among the bilateria, you can see we have this basic structure, which is that they have a head at one end and a tail at the other, and the non-bilateria don't have that. They have their sense organs toward the front, and there's some circular muscles. You have a gut that goes the whole way along. You have longitudinal muscles that would be used for movement, presumably. So that's sort of the basic body structure of bilaterian animals. And we could see that even though our bodies seem really different from the bodies of insects have a head at this end and a tail at the other. They have a gut that goes in their mouth and out the back. So that is the same sort of structure. Now, you take all the bilateria and you divide them up and you get two groups, the protostomes and the deuterostomes. So these words really just define two different groups. But the protostomes, the way you know the difference between these is that the mouth happens to form first when they're in the embryo, and in the deuterostomes, the mouth forms second, the anus forms first. So you've got this tube, and the question is, does the mouth form first or does the anus form first? And it's not like that defines what, it doesn't make the animal like what it is, but these groups differ in that sense. So you call them protostomes and deuterostomes. So we're going to be looking at insects, and insects are protostomes, and vertebrates are deuterostomes. So this is where we diverge around 600 million years ago, or maybe 550 million years ago. But whatever, let's first look at where we are on this. So deuterostomes become chordates and echidnoderms. Echidnoderms, I think, are like starfish and sea things, sort of a small group. Chordates are the main group. Chordates include all the vertebrates and also Amphioxus. But we can ignore Amphioxus, which is a kind of worm-like thing that's a pre-vertebrate. And then you get vertebrates. Oh, and you get tunicates and Amphioxus. So these are our closest relatives that are non-vertebrates, but are chordates. Chordates means they have a spinal cord. So echidnoderms don't have one. And then among vertebrates, this is where we are, also called craniates. And then we could take the diagram that we had on the previous page, and we could draw a big line down from here. We could say vertebrates become, fish, amphibians, reptiles, mammals, primates, us. That would be our lineage in there. But today we're looking at protostomes. So the protostomes divide into about three groups: mollusks, annelids. There's actually two different groups of worms, but I simplified here. So mollusks are like the octopus, the squid, the snail, the slug, the clam, things like that. You got worm-type things here, and then you have arthropods. And arthropods include insects and crustaceans and some other things. They have an exoskeleton. They have a segmented body. They have pairs of joint appendages, like wings and legs, and they have been on land for about 450 million years. So the crustaceans, of course, live in water mostly. But the insects ended up moving on land. They're among the group that moved up on land. So among all the arthropods, you have arachnids, which are spiders, myriapoda, which is many-feeted animals. These are millipedes, centipedes, maybe some other things with lots of feet. Crustaceans, over here, right, which also have an exoskeleton and segmented body, etc. And then insects. And, it's kind of surprising that insects and crustaceans are as closely related, but it seems that if you go back in time before insects became what we think of as insects evolved from a crustacean-like being, probably. And these guys, the protostomes, probably evolved from a worm-like being. That's why I said that when we went this way, we're probably a worm-like being. Our ancestors were probably a worm-like being that then diverged into either protostomes or deuterostomes. Okay, then among the insects, you have a bunch of different possible groups. We're just going to be looking at two of these today, but let's just mention them all. So you have silverfish, dragonflies, these things, which is the main focus of today: grasshopper, cricket, locust, katydid. That's all one group. Stick insects, cockroaches, termites, bugs, beetles. I always thought cockroaches were beetles, but I guess they're not, so I guess I'm confused. Beetles, ants, bees, and wasps, butterflies and moths, and then flies. And we're going to talk about flies just a tiny bit, and you'll get to see a video. So flies includes fruit flies, which we'll talk about, and also mosquitoes and other sorts of flies. I was looking up stridulation, and I found out that ants do that somewhat. We know that moths hear pretty well. Moths hear pretty well because they're prey animals for bats, and bats make a lot of sound. So moths have evolved hearing, even though they don't vocalize. But we're going to talk mostly about how grasshoppers, crickets, katydids, locusts, how they stroke one part of their body against another and produce stridulation sounds. Hearing in insects. Here's a field cricket. That's the call of the male. We'll get into more depth, but in this little picture, there's two parts. There's a chirp, and then which is called the trill. Here's just a little picture of how it works for an insect, in case you have not yet learned the birds and the bees of being an insect. Here's a male cricket. It makes that sound. It depends on the cricket species. Sometimes the females make a sound back. Sometimes they have a little gesture or a wing flap. Sometimes they just listen, but if the female likes the sound the male's making, then you could have some, well, I don't have a good euphemism for it. So I guess, yeah, these crickets are, Okay. Insects are the only invertebrates that can hear. So on that big page we had a while ago, if you took all the protostomes, none of those except for the insects seem to have evolved hearing. And insects evolved hearing at least 7 different times. Ears are always found in pairs, but not necessarily on the head. The ears can be on the legs, or the wings, or the abdomen, or the thorax, or the mouth. The ears of insects, as invertebrates, always have a localized thinning of external cuticle, something like a tympanal membrane, like tympanum membrane, or eardrum like we have. But basically it's a piece of skin that's gotten very thin and that can vibrate in sympathy with vibrations in the external world. Then there's also typically an air sac underneath that cuticle. So most fish don't have this air sac, but the air sac for some reason makes the pressure vibrations a lot more effective on the ear. But insects do have the air sac thing. And then there's some sort of a hair-like cell, like in the vertebrate ear, we do have hair cells that vibrate, which I think we've talked about. We'll probably talk about more. And they vibrate in sympathy with the pressure oscillations in the air. This is the head of a fruit fly, and you can see right in here, there's these 2 little circles and then a stem and some things going off it. And that's what you have the picture of here, these 2 circles and then a stem. And this direction, I think this thing vibrates with sound and then it moves in this way. And then maybe what's happening is that makes this whole thing move, and then that makes these cells polarize and depolarize, change how much negative or positive charge they have, and then that sends signals down the auditory nerve. And that's very similar to what's going on in vertebrates, at least at the level that I just explained. So that's what you have in insects. So then ion channels open up and close in synchrony with the vibration. And then that translates the mechanical vibration here into neural vibration, because you open and close channels that let in positive and negative charges. So the functions of hearing in insects are also the same for vertebrates. To detect other species, so for instance, if you are a prey animal of certain predators, you want to be able to detect the sound of your predators. If you are a predator animal and are looking for prey, you would love to be able to detect the sounds that your prey make. And then there's also communication, of course, within a species. So conspecific means with, right? So it's like other members of your species are called conspecifics. Members of other species are called heterospecifics. So the same mechanisms that allow you to detect prey and predators also allow you to hear your conspecifics, and that's how communication evolves. So this is why so many animals make sounds to attract mates, and they also make sounds to try to keep rivals away. So those are the 2 basic functions. There's other functions for sound, but they probably derive from these basic ones. An attractive sound that makes others want to move toward you and like you. A sound that makes others a little bit afraid of you and want to go away. And we can talk just briefly about what the meanings of sound are, sounds that insects make in here. Like all sensory systems, auditory systems have been shaped by the stimuli that carry meaning for the animals that they serve. This is a quote from some authors writing about insects. The point of this is that we tend to think that our receptors for any sensation give us information and then we learn to interpret it. But really, the way we evolve is to be able to detect certain specific types of sounds or smells or visual sights, and that basically have automatic meaning for us because we evolve a response to those things. And it's really a later evolutionary thing to have a general purpose mechanism that can perceive things that it isn't specifically evolved to detect. That might not make tons of sense right now, but it's something that will gradually become clearer over the course. Within that, you have this notion of categorical perception, or recognizing a specific meaning for a thing. And here's another quote. Responses to conspecific signals, right, the communication sounds made by others of the same species, are usually evoked only by stimuli that meet more or less stringent structural criteria. In other words, conspecific signals usually must be recognized as such before they elicit behavioral responses. So an example of this, we'll see it with crickets in this presentation, but an example would be, let's say, the vowel ah and the vowel o. If I were to try to do something in between, so ah, o, ah, ah, and I said, is that an ah or an o? You'd say ah. You're not really sure, you're not sure which category to put it in, and if it's not in a category, with speech at least, you can't do much with it. You just have to say, I don't know what that was. So that is an example that would apply more generally. We're going to see that insects and all different kinds of animals have very specific frequencies that they signal at, like pitches, very specific timing that they use. And if the timing is altered just a tiny bit, it often doesn't just reduce the likelihood of the right response to it. It'll just make it so that the other animal doesn't really perceive it as being the right kind of call at all. So that's the idea of categorical perception. And then something very interesting just to think about is that differently tuned receptors allow for different channels of communication or detection. So let's say you have, in the cricket call that we saw before, there's the chirp and the trill. You have pulses fairly close together, a lot of them, or you have just two at a time and then space and two at a time. And having a receptor for more than two in a row versus just two and then a space ends up giving a cue as to whether the sound is an aggressive sound or an attractive sound, courtship sound or a territorial sound. That's a very, very, very simple change in the sonic structure of the signal, but it can have two different categorical meanings. And there is some habituation to repeated or familiar stimuli, meaning that there's some rudimentary form of memory. I'm not going to go into this right now, but basically if, let's say you played a recording for an insect of another insect calling, and at first it liked it, but you kept playing the exact same recording in the exact same way, the insect may habituate to it, meaning it would be less sensitive, less responsive to it after hearing it again for a lot of time. So then just briefly a comparison of hearing in insects and vertebrates. I found this pretty interesting, and I mentioned in a previous video that most animals, including insects, are sensitive to sound within the range of 2 to 15 kilohertz. So that is pretty high, but it's irrespective of body size. So from elephants all the way to the tiniest insects, pretty much everybody can hear in that range. And then some beings can hear much lower than that. Elephants can hear lower than we can. And then some animals can hear much higher than 15 kilohertz. But there is a kind of common range that virtually all animals can hear and use in their vocalization. We will talk about these terms a little bit more later, but I'm just going to introduce them now, the idea of amplitude modulation and frequency modulation, called AM and FM. And that is related to radio signals, AM and FM radio, but not obviously, so you don't have to try to map that onto that understanding just yet. AM, amplitude modulation, simply means that you're modulating the amplitude, so you're modulating the volume level of a signal. So if you have is your carrier, you can amplitude modulate its volume by going or right? Those are amplitude modulations. You could do it fast or slow. Okay. Anyway, you could do the same thing with frequency. So if my carrier is again, I can go and that's modulating the frequency. And you could do it fast, or slow. Okay, amplitude modulation is common in insect communication, and it's detected by most insects. And FM, interestingly enough, is not common in communication. Animals seem to not have the ability to do frequency modulation, actually, but we do see one animal that does that. But it's detected by many species because predators like bats and birds do modulate the frequency of their sounds. So animals might evolve the ability to hear something that they don't actually use in their communication sounds. And a last thing is that in some insects, you find tonotopic organization. In the last lecture, we talked about how the basilar membrane is tonotopically organized. Tone is obviously frequency, and topical is like a mapping in space or shape. So the one end of the basilar membrane responds to low frequencies, and the other end responds to high frequencies. And so it's tonotopically organized, and many of the nuclei in the brain are organized that way too. So on the foreleg of crickets that has an ear on it, one part of that ear, one part of the leg is more responsive to low frequencies, one part's more responsive to high frequencies. So the ears are made in different ways and they appear in different parts of the body, but they might still obey these same sorts of organization, like tonotopic organization. Let's look at an example of a field cricket. And phonotaxis, it sounds fancy, but it's not super fancy. You're going to learn about it more next week when we talk about fish, but I'm going to introduce the term now. Phonotaxis simply means moving relative to a sound source. So phono is sound, and taxis is movement. Technically, you say positive phonotaxis or negative phonotaxis. Positive phonotaxis means moving toward an attractive sound, such as a mating call. Negative phonotaxis means moving away from a repellent sound, such as an aggressive call. But often people just use the term phonotaxis in the positive sense, meaning moving toward a sound. So, as an example, we're looking at the field cricket, and you have a little picture at the upper right. And the field cricket can hear all the way up to 100 kilohertz. We can hear only up to about 20 kilohertz. So there's 3 or so more octaves in there, 20 up to 40, 40 up to 80. Yeah, so more than 2 octaves above what we can hear. Their calling song is at roughly around 5 kilohertz, 5,000 hertz. And we did hear that before, and we'll probably hear it again. And as long as the calling song is at the 5 kilohertz level, a female, if she's interested in males at that moment, she'll hop toward the caller making the 5 kilohertz sound. If you raise the frequency, did it with a higher pitch, she's still going to hop toward it until it gets really high at 2 octaves above 5 kilohertz. Then at that point, it's unpleasant for her and she's not going to hop toward it anymore. She will hop away from it. So that's negative phonotaxis. And the authors that I'm reading on this wrote that those are categorically different behaviors. They're not variants. It's not a graded variant of the same behavior. She's really responding to those 2 different songs in 2 totally different ways and opposite ways. So some species of cricket have different songs, like a calling song if they're from far away and a courtship song if they're at a closer distance. The courtship song is more difficult and is hence a better indicator of mate quality, but the calling song from farther away simply lets the female know about what species the caller is. Some species, and this is definitely the case for the field cricket, for T. oceanicus, have one part of the song that deters rival males while a second part attracts the females. So in this case, the chirp is the attractive thing. And it's just the single syllables but widely spaced. And then the trill, double chirps. And faster overall, that thing is going to be repellent, whereas the chirp is attractive. So a single male is going to be singing a series of these, some chirps, some trills, some chirps, some trills, but that has the effect of both being repellent to rivals and attractive to females. And then it's important to note that the distinction between the chirp and the trill is dependent on the timing only, not on the frequency or timbre. So if you looked at each one of these individual pulses, in the chirp and in the trill, they're going to look the same. So it's just the timing of them that changes. And that is this fascinating concept that it's much easier for an animal to change the timing at which it produces certain sounds than it is to just make new sounds altogether, to make the same sound at a different frequency or to make the same sound with a different timbre. So in music, we're always talking about different pitches and different timbres and everything like that. But a lot of animal vocalization happens just with one noise-making mechanism. And you can do a lot with that. You can do a lot with a single noisemaker in the timing domain, but not so much in the frequency or timbral domain. And that's something we'll come back to. I mean, in a sense, like we make all of our music out of just 12 pitches, right? We do it in different octaves. We have different instruments playing. We get louder and softer, right, with crescendo and diminuendo. But we do just use those 12 notes in a sense. And the same with speech. We can say just about anything using about 20 different phonemes, or maybe it's less. It might be even like 15 different phonemes. So in a sense, we do the same thing. Okay, here is T. OCEANICUS. That was full speed. Here it is at half speed. This time we can see there's some 2s and 3s, and then in here is a series of individuals next to each other. Now I'm going to go to quarter speed. There you can really hear the 2s and 3s. Now the individuals next to each other. 2s. And we're going to go even slower. This is 1/8 of the speed. It really starts to sound like an owl. You can hear that there's some frequency modulation. Right, so descending within each pulse. And now, I believe it's 1/16 the speed. And we'll zoom in a bit. And in this is from the Raven viewer that grad students have been using. What you're seeing up here, this dimension is how loud, how much pressure there is. So that's amplitude. So from here up to there is the amount of pressure. I'm not sure what these units are, but this is 10 somethings, KU. And this is time. So this is 0.1 seconds, 0.2, 0.3. So this is 1 second of time. And we've just slowed it down. So this actually takes us 16 seconds to get through. Then down here you have the spectrogram. So this one up here is pressure or volume. This one are frequencies. And so these are in kilohertz. So this is 1 kilohertz, 2 kilohertz, 3 hertz. So 5 kilohertz. So we can see in here that this starts at around 4.5 kilohertz and descends to about 4 kilohertz. And then there's also a harmonic up here. This is 4.5, the first harmonic, so double it is 9. So this is just under 4.5. This is just under 9 kilohertz. So we can also go a bit more in depth. The duration of each one of these things is about 50 milliseconds. I'm figuring that out because this is 1/10 of a second, 2/10 of a second, right? So 1/10 of a second is 100 milliseconds, and then this is about half of 100 milliseconds. So I'm calling that 50 milliseconds. That's the pulse duration. The pulse pitch we saw was about 4.5 to 4. It's 4.5 and it's descending like, whoo, right? So that descent there from around 4.5 to 4 kilohertz. You could measure instead the interpulse interval, so from the beginning of one pulse to the beginning of the other, and that's about 70 milliseconds. So you could look at the space in between them is probably about 20. So 50 plus 20 is about 70 milliseconds. And then you could also look at the duration of these two together. If the two together is a chirp, then so the duration of before the next thing is about 200 milliseconds. Those are all different things that you might be looking at. So we're going to talk briefly about duets. The one main thing I want to say about this is in literature about animals vocalizing and singing, there's definitely a way more focus on what males do and what callers do. There's very little understanding of what listeners do that makes them like a certain sound or not. And there's much less understanding of what females do. So, as science matures, scientists are starting to really figure out that a lot of the most fascinating stuff is what happens in females in any species. And in birds, for instance, I was just reading an article where they're saying that it's always assumed that males evolved singing first, and that in some species, females developed singing too, but they developed it from the males. But turns out that's not right. In all birds, there's 5,000 species of birds that sing, I believe. And the way it evolved is in both males and females. And it just so happens that most of the species that scientists study, it's just the males that sing. But that's kind of because they're easier to study. So as we go along in this class, there is a lot of focus on what the males of a species do. But I want to make sure that first of all, scientists don't understand completely all the things that are most interesting. They're often looking for stuff they already understand. So there is much more to learn. And the cutting edge science is really discovering some really brilliant stuff about the interactions between males and females, and interactions between males and males, females and females, males and females when another male is there or when another female is there. I mean, it's all kind of fascinating things. Okay, so let's talk about duetting just a little bit. Many different species of crickets do duets, so male-female duets. The male would sing, the female often sings back, and then if the female sings back, then the male hops toward her and sings again, and then they repeat the little thing until they are mating. And then there's going to be differences between the male song and the female song, so if you acoustically heard song of one of these crickets, it would still be the same song. You could hear it's the same species song, but there's some subtle differences. So the rate of the onset of the syllable, I drew a little red bar here to show, like, if it's very sudden, it's ah. If it goes slowly, it's ah. So this one has a sort of a gradual onset. The amount of space between the syllables, I'm just calling this a syllable. It's the same thing as a pulse. The duration of the syllable itself, I called this the pulse length before, and I called from here to here the interpulse interval. So if you could add this and this, that would give you the interpulse interval. The intensity of the syllables, so that's like how loud this thing is. It's going up to about there. This is at its most intense point. And then the spectrum of the syllables. So I took a picture of the spectrum for that syllable we were looking at. And this is from 0 hertz to 10 kilohertz. So 10,000 hertz. And this is the one we were looking at before. Most of the activity is around 4.5 to 4 kilohertz, sloping down. And then there's the second harmonic. Now we're going to look more specifically at how the whole stridulation thing works. And this is a mole cricket. I thought that everybody who plays a horn would be pretty interested in this, because the mole cricket, you can see it's got these little things on its hands. It digs a hole like this and makes it really smooth on the sides. Maybe it uses saliva or some other excreted liquid to make these sides really smooth. But it kind of creates a perfect acoustic atmosphere and horns that amplify the sound that it makes. And these things are tuned to the tone that it produces. So it's really pretty fascinating. You could apparently hear it from somewhat far away. This is a side view. If you cut into the ground, you could see the little cricket down there. He's facing backwards. So this is his head there, the wings back there, and the wings are going to produce a sound that goes up there. And we can hear it. So all these mole crickets are Grillotalpa, the genus, and this is a Grillotalpa vinae. I listened to some of the other ones, but man, listening to some of the other songs, they're really painful to listen to. But this one I thought was the nicest sound. Okay, let's see how it does it. Here is another video, and I'm going to do the same thing where I play it at normal speed and then slow it down. Okay, it's normal speed, and we can see frequency-wise it's around 7 kilohertz is the main sound. And then there's a harmonic up there at 14 kilohertz. Here it is at half speed. And since at half speed, it's also probably about 3.5 hertz. This is quarter speed, so this is probably going to be 1.75 kilohertz. And we can now start to hear the individual pulses. Okay, let's go to 1/8 of the speed. Okay, so we're hearing that probably about 1,000 hertz, and you can definitely hear the individual pulses. We're going to now 1/16 of the speed, so this should be about 500 hertz. Like 1/16 of 7.5. Okay, so there is really clear that we're hearing a series of pulses, individual pulses. And now we're going to slow it down even more. We can even count up how many of these pulses are there from, let's say, 60 milliseconds to 160. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. About 12 per 100 milliseconds. That's 120 per second. So 120 hertz. Those pulses are happening at about 120 hertz. And then within each pulse, the pulse is at 7 kilohertz. Okay, so let's see how the mole cricket does it. How do they do it? This is the underside of the wing of a male Grillotalpa bene. It's hard to imagine this, but see if you can. This is the right wing. So this is the back of the animal, and this is the front of the animal. So this is the wing. The muscles control this part of the wing. This is a sort of flexible membrane. And then there's these things on here, which we'll get a better look at in a second. But this on one wing is rubbed against this thing, the plectrum of another wing. And that produces a sound. And then this thing in here is the harp, which is just a membrane that is going to go up and down with each of these clicks. These darker regions in here are kind of flexible. The CU2 vein is also called the pars stridens. It's stippled on the bottom. It's got like little bumps on it. The plectrum on the other wing, so this is just one of the wings, but the plectrum on one wing rubs against the stippling on the other wing, and that produces the stridulation. That's what we said is like a violin bow in a previous presentation. You can see close up, if you really looked at that vein, you'd see there's these bumps on it. And if you looked at it from the side, you'd see the ridges. So you're running the plectrum, and then that's pretty fun. And they're more widely spaced here and more closely spaced here, so that would produce a higher frequency here and lower frequency here. So in the previous animal we were looking at where there's a drop in the tone over the course of each pulse, it could be because of something like that, something structural. The area at the center of CU1, this thing roughly, is called the harp. It resonates with and amplifies the frequency produced by the stridulation. Stridulation is rubbing the plectrum against these stubble. And here's a picture of the wing. So you'd have to flip this around to make it look like this, but that doesn't matter. The harp is just this membrane here. It really looks like the sail on a sailboat in a sense. And that's just going to sort of move back and forth with the vibration, sort of like the cone on a speaker. I'm getting these numbers from this article by Bennett Clark, and this is a lot lower than what we were seeing in our diagram. But in this example, the fundamental frequency is at 3.4 hertz, and that activity at 3.4 hertz, 3,400 times a second, that produces a harmonic, just like on a violin or guitar string or any instrument, at twice 3.4, so that's 6.8. And then it produces a harmonic at 3 times that, so that ends up being 10.2 kilohertz. Just another little picture. I think this is pretty fascinating. If you make the cricket very cold, it basically can't move its muscles fast enough to produce the stipulation sound. So it just goes a And earlier in the evening, before the cricket has warmed up, it makes sounds kind of like that. And as it starts singing more, it gets warmer and it can do this better. Then at slightly warmer temperature, you can see now it's able to produce its sound, but there's a lot of space in between the little pulses. And then as it gets warmer and warmer, at C and D at the warmest temperature, 16 degrees here, Celsius, each individual pulse is longer. So this one slightly longer than this and longer than this, definitely. So these pulses get longer, and also the space between the pulses gets smaller. So it produces much more of a consistent tone. And then what we can see here is the pulse tends to last about 8 milliseconds at this later time. Here it might be 6 milliseconds. The interpulse interval from here to about there is about 14 milliseconds. We just know this because this is 40 milliseconds. And also I got these numbers he told me this in a different part of his paper. And then the frequency within the pulse, if you divided these up, there'd be a lot of really fast notes. It would be 3,400 times per second in there. So it produces a high-pitched tone. So you really have two frequencies here. You have within the pulse, and that's a very high-pitched and then you're doing the tones. You're going, so the wing is going, and you put them close enough, and you get this sort of like double sound. You get the tone that's in here, but then it has basically an amplitude modulation at this slower rate. Summary. Many species of insects sing, and even more species of insects hear. Insects are the only invertebrates that hear and sing. So they evolve these abilities independently from vertebrates. And you call that convergent evolution. So anytime there's a trait that's shared by different groups, you could say, oh, did they both have it because an ancestor a shared ancestor had that thing? Or did they both end up figuring out the same trick at different times by different means? So that's what you have here. Insects and vertebrates figured out how to hear. And with insects, the 7 different times they figured out how to hear. But they do it in typically a very similar way. So that suggests that hearing and singing are very valuable. And even the way that they do it is maybe like one of the best ways to do it. Ears have common properties across all animals. And this is the convergent evolution thing. It's not because the shared ancestor had all these things. A resonant cuticle, like a skin type thing, a tympanum, that opens and closes ion channels. So with the little air pocket and the channels that open and close because the tympanum is moving back and forth. Ears are always going to be in pairs, and they're always sensitive to frequencies, somewhere between 2 and 15 hertz. And they're sometimes tonotopically organized. But ears also have some differences across species, so they could be on any part of the body. And one more screen of summary. Orthoptera species, so this is the specific type of insect we've been talking about here, grasshoppers, crickets, katydids, cicadas, produce sounds through stridulation. Stridulation is rubbing one part of the body against another. It could be a leg or a wing. It could be something else, but all the examples that we saw are rubbing either a leg over something, which is what grasshoppers do, or rubbing wings against each other, which is what crickets and I think katydids do. So the mole cricket that we looked at, the one that's in the hole, has a jagged tooth-like vein that was like a CO2 or something like that, which was rubbed against the plectrum of the other wing. And then it had a harp in the middle of the wing, which is sort of like the sail on a boat, but it goes up and down like the cone of an electronic speaker. The harp portion of the wing is like the cone of the speaker that creates the pressure fluctuations. And then the hole dug by the cricket amplifies that sound. So flies and mosquitoes, we didn't really talk about these, but you're going to see a video of mosquitoes doing stuff. Flies and mosquitoes produce tones according to the wing speed that they're using. And fruit flies, Drosophila, that's a famous species. It's one of the best studied species of all. They alternate between pulses and more sign-like tones. So I might show you a video of that, but it basically sounds like so that's called the courtship song. I think if the male doesn't do that, then the female does not want to continue with the courtship. And some scientists that I was reading try to figure out when is First of all, the courtship song is not totally innate. It's complex. So the male is trying to figure out what the female is doing and then adjusts his courtship song based on that. And whenever he's kind of far away from her, he goes, brrr. And when he's closer, he goes, er. So it seems like based on her behavior, his courtship song changes. And if he does it right, then she's interested in mating. Now that's just a solo song, but mosquitoes are totally fascinating. So you'll see a video that does show you what's going on, but mosquitoes tend to harmonize in duets at intervals, such as the perfect fifth. And so the female wing speed is a little bit slower than the male wing speed. But then as they're courting, it seems like they're actively adjusting their wing speeds to produce a pure interval, usually at the fifth. Sometimes it could be at the octave. And in the video that you're going to watch, I can hear some other intervals in there. So I'd be curious what you think about that video. And then finally, duetting and communication between males and females is probably much more common than currently recognized. And we will get to see more of that as this class goes on. But as I mentioned, scientists tend to study things that they already understand. I know that sounds like I'm saying something bad about science, but the way science works is it's often so expensive to do studies, and you have to apply for grants, and to apply for a grant, you have to say, we want to do this thing, and we're pretty sure we're going to get these results. And they often tend to keep studying the same things in the same ways. So science has almost always focused on the sounds made by singers in the different species we're talking about. But really, the reception, the response to those sounds is super, super fascinating. It's what we do as listeners. You know, we're all musicians. We like to make sounds, but we like to make sounds because we have listened to them and we like the way it sounds when we listen to them. And when we're playing them, we like the way it sounds when we're listening to ourselves playing, right? Listening is super complex. Liking what you hear, super complex. And in a sense, the actual making of the sound is not really that complex. I don't want you to take from this kind of discussion that there's some sort of a male bias in what's interesting about what animals do or what's complex. There is a male bias in the science at the moment, but we're going to keep seeing some really fascinating things that deepen that picture and help us understand what humans do, because humans are totally fascinating when it comes to all the ways that music is used by everybody in human culture in just all kinds of fascinating ways. So that's sort of the trajectory of where we're going with duetting and communication and all kinds of things. And then finally, as you'll see in your reading, you're going to read 2 chapters from Bug Music by David Rothenberg, and there's some cool dance-like type displays as well. And I hope you enjoy this week's worth of activities.